Hi everyone, my name is Doyon Laliberté and I'm a PhD student currently working under the supervision of Dr. Roger and Dr. Poudrier. Today I'm going to present to you a small facet of my project on the B regulatory B cells in the context of HIV. As another chronic infection, HIV is associated with exhaustion. It is now well published that there is an increase of the induced Treg frequencies, and the effector and memory cells lose their capacity to proliferate upon stimulation. Also, they will increase the expression of immune checkpoints such as PD-1. Altogether, those changes lead to an impaired antiviral response and the inability to eliminate the virus. Even if most of those studies have been done on T cells, I will show you that it is also true for B cells population. The human marginal zone B cells are a CD1 plus innate B cell subset. We identified them with these markers and their precursors, that we called precursor-like marginal zone B cells, possess also the maturity marker CD10. Marginal zone B cells possess a polyreactive BCR and can differentiate in short-lived plasmacytes with no memory. This means that they can produce polyreactive antibodies, mostly IgM, upon activation via their BCR and Toll-like receptors. Those cells use therefore T-independent activation. They can be found in the circumference of the germinal centers in secondary lymphoid organs. Differently from the mouse, human marginal zone B cells have the capacity to recirculate in the blood. They are important for the trafficking of antigens to the follicular dendritic cells and follicular T cells. Marginal zone B cells require B cell activating factor, BAF, for their selection and survival. Also, BAF can contribute to their class switch recombination. We have also previously shown that they have a BREG potential. It is important to know that there is no exclusive BREG subset in human comparatively to the Tregs. Almost all B cell subsets have the capacity to produce IL-10 upon stimulation, which is why it is difficult to target only one BREG subset. In the literature, CD39 and CD73 have been associated with a BREG potential. Some studies say that those are true BREG markers. Also, the NR4A family is essential for the maintenance of FOXP3 in Tregs, and they control the expression of the immunoregulatory protein CD83. We have previously shown that the precursor-like marginal zone B cells of healthy individuals highly express each one of these molecules, as we can see over here. Their expression is similar to CD19, a B cell marker which is strongly expressed by those cells. They are also the subset that express higher levels of NR4A1, NR4A3, and CD83 comparatively to mature marginal zone B cells and total B cells, as we can see. We have also shown that precursor-like marginal zone B cells have the capacity to control CD4 T cells proliferation in vitro, and that it is dependent of the CD83. Those results show that precursor-like marginal zone B cells have an important BREG potential. Also, we have previously shown that in the blood of progressors from the Montreal Primo infection HIV cohort of the FRQS, there is an excess of BAF, an increase of the frequencies of precursor-like marginal zone B cells, an increase of IL-10 positive precursor-like marginal zone B cells, and that their BREG phenotype is altered. The age-associated B cells have been found in both mice and humans living with chronic infections or autoimmune disease such as SLE. They are known for their high expression of CD11C and TBET. It has also been described that those cells involve TLR and IL-21 signaling. They are also associated with the production of autoreactive antibodies. To better understand the dysregulation of precursor-like marginal zone B cells in the context of HIV, we did a transcriptome analysis by RNA-seq of cell-sorted precursor-like marginal zone B cells of the blood of progressors. Here we can see that CD22 and CD72, which are BCR negative regulators, are significantly more expressed by precursor-like marginal zone B cells from the HIV-infected individuals than the healthy individuals. Those results suggest higher activation levels at baseline in those HIV-infected individuals. We also look at the exhaustion markers FcRLA5, which is an IgG receptor that is upregulated in other chronic infections like HCV, and CD5J, which is an immune checkpoint associated with the B-cell exhaustion in chronic graft versus host disease. The results show a significant increase of mRNA levels in precursor-like marginal zone B-cells from progressors compared to healthy individuals for both of these markers. After that, we wanted to know if our precursor-like marginal zone B-cells shared some markers with the age-associated B-cells. We found that precursor-like marginal zone B-cells from HIV-infected individuals express higher level of CD11C compared to the healthy individuals. They also tend to upregulate T-bet expression. Those results show that precursor-like marginal zone B-cells share an exhaustion profile similar to age-associated B-cells in the context of HIV infection. Then, we looked at the expression of TLR7, which can recognize single-strand RNA, and TLR10 that is involved in the recognition of structural proteins of HIV, and we found that their gene transcripts are higher in precursor-like marginal zone B-cells of the progressors compared to the healthy individuals, and this for both of these proteins. Also, those cells express higher levels of EL21 receptors than mature marginal zone B cells. Those results mean that they are more susceptible to share similarities with age-associated B cells, since IL-21 signaling is involved in the differentiation of this population. In conclusion, these data suggest that precursor-like marginal zone B cells possess an exhaustion profile similar to the age-associated B cells. These cells could contribute to autoimmune manifestations and hyperglobulinemia observed in HIV or other chronic infections. Finally, I would like to thank my principal investigators, Dr. Roger and Dr. Poudrier, and all my colleagues for their support and their help on this project. I would also like to thank all the other persons that participated or shared their expertise for the success of this project. Also, I would like to thank you all for being here today and for your attention.